This program is a presentation of UCTV for educational and non-commercial use only.
So what I'm going to talk about today is entitled the Promise and Challenge of Personalized Cancer Therapy. And what I'm going to hopefully convey to you is the incredible sense of excitement that exists within the cancer research community and also the clinical cancer research community, the people that actually deliver care to patients, about how our understanding of the molecular wiring of the cancer cell is turning into new and improved therapies that are making a significant difference in the lives of people with cancer. Now, in telling you this story, which I think is actually a triumph of modern science and the translation of modern science into medical applications, this does not mean to say that we've conquered cancer. In fact, we've really only put our foot on the first rung of the approach, or of learning the approaches to targeting and curing cancer. But I think that the work that I'm going to describe to you today tells us about how to approach cancer as a disease. It's no longer a monolithic disease. Cancer is not any one disease. Many of you, most of you I'm sure will know, that cancer can arise from maybe 200 different locations in the body, and each one of those cancers can be quite distinct. But even if you take a disease like breast cancer, which is a remarkably common disease in our society, maybe 150,000 new breast cancer diagnoses, what we've come to understand is that there are large subsets of breast cancer. So breast cancer is not just a monolithic disease. There are maybe 5 different subsets of breast cancer. And each of those subsets of breast cancer should be and are being treated rather differently. But then when you get down to the details of the individual patient, what we've also come to understand is that each patient's tumour can be quite distinct in much the same way that each individual human being is distinct. And so what we're trying to understand then is how can we apply tools that will allow us as basic scientists or as clinical physicians to deliver the best cancer care to individuals with the disease that takes full account of both the specifics of their individual tumour and the specifics of them as a person. This presents enormous challenges, as you can imagine, but at the same time, we've now developed this understanding of cancer as being quite a unique set of individual types of diseases that need to be tackled on a personalised basis for the best possible care. So, as my mother likes to tell me, I am not a proper doctor. So the advice that I can give you today is based on the fact that I'm a PhD, and I've spent a lot of time talking to people who are proper doctors, the type my mum would rather I had been. Most of my patients have 4 legs, fur, and a tail, and that's in part because my lab hopes that by studying cancer in genetically engineered mouse models, that we can inform our understanding of the basic processes that drive cancer, and also understand new ways to treat cancer in an experimental system that can then be more rapidly translated into the clinical approaches for treating patients with cancer. So I couch anything I might say that might sound like medical advice should be taken with the hefty pinch of PhD salt that I bring to this lecture. Okay? So, the promise of personalized cancer medicine. Ultimately, personalized cancer medicine seeks to improve the cancer care for all patients. So you think about it, all cancer is personal, right? Everyone, when you get cancer, it's a personal diagnosis, a personal shock, and a personal challenge. And each person's cancer, even if it's something like a common disease, will still be treated in an individualized way. But what we seek to do is to bring to bear new tools that allow us to diagnose cancer, especially earlier in its course, and to predict its course in the clinic so that we can apply more accurate and sophisticated tools of treatment to give cancer patients the best possible outcome, which, as far as we're concerned, is a long, healthy life. Now, coping with the cancer burden in society may not necessarily mean curing cancer. Right? It would be really fantastic if we could come up with the magic, scientific, validated tools that would eradicate cancer in a person's body. However, I think we have to be realistic, and what initially we're aiming for is the control of cancer, so that the cancer in your body is controlled and is not a major source of morbidity and influence in your life, that you can live a perfectly happy life, even though you have to take a medication for cancer. That's the first step, and then that will be, I think, a step on the ladder to actually being able to cure it. And I'll talk about a very specific example of that a little bit later on. What we also want is the generation of better and safer cancer therapies that are tailored to the specifics of a patient. If a person is diagnosed with breast cancer and they have a particular form of breast cancer, then we want to give them a drug that will target that specific breast cancer. Whereas the old school way of doing things was treat the disease as a monolithic thing and give everyone the same drug. That appealed to the drug companies because it meant they could sell lots of drugs to lots of patients. But it was also a very simple but rather one-size-fits-all type of approach to the disease. In addition, we'd like to be able to minimize treatment protocols that have been proven not to work for specific subsets of patients. And again, these diagnostic and prognostic tools that we have available to us and that are being developed are now allowing us to be able to say to individuals or groups of patients, this is a therapy that we know is going to work and provide you benefit. But this is a separate therapy, the same therapy to a different subset of patients is something that we know will not work. And therefore, we'll think about an alternative approach that might be more specific to the type of cancer that you have. Now, the promises of cancer medicine are also counterbalanced to some extent by the challenges of personalized cancer medicine. So first off, we need to make these tools. They don't just magically appear in the lab, and they have to be tested in an academically and scientifically rigorous fashion, so that if a clinician makes a prediction about a disease in a patient, that they can provide the patient accurate advice that is more likely than not to be absolutely correct. And there are very strict tolerances about the way these tests might work, which are enforced by the FDA, so that if you actually make a prediction about how someone's disease should be treated, that you're providing them the best possible medical evidence based on a strong statistical probability that you're giving them advice that's relevant and important to their disease. The back end of this is to generate, evaluate, and optimize the treatment of new cancer therapies designed to treat specific subsets of cancer patients. Now, many of you, I'm sure, will be familiar with the system by which a cancer drug gets tested and approved. Initially, in a phase 1 clinical trial, the drug is tested until you find a dose that is tolerable, but is usually the maximum or close to the maximum tolerated dose. And those phase 1 clinical trials, generally speaking, are not designed to test efficacy, to actually see evidence of tumor shrinkage. In a phase 2 clinical trial, then you start to target specific types of disease, breast cancer or lung cancer or melanoma. And then the phase 3 clinical trial, which is the one that people often refer to as being the pivotal piece of the trial, is when you conduct a randomized, double-blind, placebo-controlled study of your new therapy either against a placebo or against a pre-existing standard of care. And there's and that's the thing that you would then get submitted to the FDA, and the company says, will you approve us to sell this drug to cancer patients? And there's a natural tendency to think of that's the end of the story. The drug's approved, presto fixo, off into the clinic and off you go. And the reality is that approval from the FDA is just the beginning of trying to understand how that drug works. You're simply demonstrating that the drug has clinically relevant, statistically significant benefit in patients, and the FDA will approve its use to be given to patients with a specific type of disease. But the reality is that that's just the beginning of the process. Because the question is, once a drug has been approved as a single agent, what can you combine it with to make it better? What's the best schedule of that drug? The schedule that was used in the clinical trials might be effective, but it might not be the best schedule. What subsets of individuals respond better or worse to this drug? And so the phase 3 clinical trial is by no means the end. It really is the beginning. It's the beginning of the process of trying to work out how best to use a new cancer drug in the most appropriate way. As I said, at the moment, not all types of cancer have personalised treatment options, and that's partly because we are still in the exploratory phase of trying to work out the molecular genetics and the cellular wiring diagram that I'll come on to in a minute of how cancer works for each and every one of the over 200 types of cancer that there are. There has traditionally been some resistance in the private sector to move away from the one-size-fits-all approach, in part because if you have a drug that's approved for breast cancer patients, you would like that, as a company, to be given to as many breast cancer patients as possible, because that will maximize your profit. What we've come to understand, though, is that even drugs that are unique and relevant to a specific subset, even for a relatively niche disease, can end up being extremely successful drugs, so that it fulfills the company's mandate and mission to make new medicines, but it also fulfills their mandate and mission to be profitable to their investors. And I hate to say it, the reality is of cancer drug discovery and drug discovery in any disease is it's absolutely reliant upon a healthy, vibrant private sector that are willing to go off and make the investment in making drugs, because universities just don't have the wherewithal to become drug companies. And then the costs of new drug development are high, and health insurance reimbursements remain uncertain. And of course, one could spend an entire week's worth of lectures on that last bullet point, and I shall not go there. So the numbers of the statistics of cancer are sobering. And I'm sure all of us in this room have either been directly affected by cancer or have a member of our family who has had cancer or a cancer diagnosis. Because about 25% of all US citizens will be afflicted with cancer in their lifetime. In 1993, there were approximately just over 1.1 million cases of cancer diagnosed. I guess it's time for me to update this slide since it's now 16 years old. But one of the good things that we've seen, and maybe you've seen this in the press recently, is that having for years and years and years seen increasing numbers of cancer deaths in the United States, we've reached an inflection point. And in fact, the numbers of cancer deaths on an age-adjusted basis in the United States are actually starting to go down for cancer. And that's actually a remarkable thing. There were many people that predicted that might never happen. Now, it's unclear why that has happened, and there are armies of statisticians and scientists trying to understand how that works. Undoubtedly, some of it is to do with smoking cessation, because as many of you will know, the number one killer of cancer in our society is largely lung cancer, and that's obviously largely to do with smoking. Not always, but largely. And there are many other types of cancer which are associated with the risks associated with tobacco use. But it's not necessarily clear that that's the sole answer to the inflection point, but it certainly is very good news. And as I said, the numbers of cancer deaths now are starting to show signs that we're making some progress. These are estimated cancer deaths. So you can see here, lung cancer really does sit at the top of the tree here in terms of its prevalence and also in terms of its lethality. I think the other thing about lung cancer is it has a very aggressive course from the time that it's diagnosed. But as you'll see, there's hope for lung cancer because we're starting to understand some of the subsets of this disease. Obviously, in women, breast cancer is a major concern, 150,000 new diagnoses. Colon cancer also very significant, and you can read the list here. Some of these we're making great progress again, but there's a couple of diseases that seem, for reasons that which I'll be happy to go into, where we've really not made any progress. The number one that we have not made progress against is pancreatic cancer. This remains an incredibly difficult disease to crack, whereas we're starting to make progress against really intractable diseases that have been extremely difficult in the past. I'll touch on the story of melanoma very shortly. Pancreas cancer remains a remarkably difficult disease to diagnose and to treat. And the other one that's particularly difficult to diagnose and treat is a brain tumor known as glioblastoma multiforme. In 1971, President Nixon tasked the National Cancer Institute, which was a pre-existing arm within the National Institutes of Health, to tackle the problem of cancer and to aim towards the goal of eradicating cancer in as short a possible time. And there's a fantastic backstory to how this happened. There's a fellow by the name of Siddhartha Mukherjee who wrote a book recently which was published that describes the war on cancer from the perspective of a physician scientist working at the Dana-Farber, and described how some of the early chemotherapeutics were discovered. When this work was started in 1971, the roots of cancer were unknown. There were many different arguments and discussions about where cancer might come from. a variety of different hypotheses. One of the most favored hypotheses, that there was somehow a viral etiology, that cancer was somehow infectious. We've come to understand that while viruses can play a role in the initiation of the cancer cell, cancer as such is largely not a contagious disease. But I'm sure many of you will be familiar with the fact that, say, human papillomavirus is the initiating event in the genesis of many cervical cancers. If you have an HPV infection, it doesn't mean you're going to get cervical cancer, but for that 1% of women who do get cervical cancer, HPV appears to be the initiating force. Similar with hepatitis viruses, the chronic inflammation created in the liver as a consequence of consistent viremia is the underlying, or can be an underlying reason for the development of liver cancer. Again, an infection with hepatitis virus does not mean that you will get liver cancer, it just means that you are at higher risk for contracting the disease. But coming back to this, is that in the course of the past 40 years, what we've now come to understand is that cancer is a disease that rises as a consequence of genetic alterations in key genes that are normally involved in the normal processes of human development and tissue homeostasis and our normal physiology. And it's when these genes are damaged, often by mutation, that the cells of a cancer acquire the capacity for independent, unrestrained cell proliferation. And there are many hallmarks of cancer, but it's the proliferation that tends to drive the growth of the primary tumor. And then the thing that's particularly lethal about cancer as a disease, and this applies pretty much to most or all cancers, is the capacity to pick up from one site in the body and take up residence in a brand new site in this process that's known as metastasis, which is generally out of the reach of surgical technique. And so it's the process of metastasis that makes cancer such a lethal disease. If cancer was only restricted to the organ from which it arose, chances are surgeons could cure it 9 times out of 10. And it's the metastatic process that has been particularly difficult to deal with. Now, every single one of us in this room, and I can say this without any fear of contradiction, started as a single cell. This is the ultimate stem cell, right? The fertilized human oocyte is totipotent, and it gives rise to every single cell in our bodies, every lineage, and has this remarkable capacity to give rise to all the lineages of the human body. And it does so based on a set of instructions carried in DNA in the nucleus, arranged in 46 chromosomes, and it's estimated, although the number's a little still soft, that there may be, actually this is probably an overestimate, probably 20 to 25,000 genes. And the typical human adult has maybe 100 billion. Now that's an American billion, so that's 10 with 14 zeros after the back, okay? Unlike a British billion, but I won't go there. And in the course of a day, millions, if not billions, of our cells turn over in a regular, orderly process. Our skin, our blood, the lining of our gut, all sorts of normal processes of cellular replacement in order to rejuvenate the tissues that are often in the face of particular environmental insults, like skin exposed to UV light. I was out playing soccer in Marin on the weekend and got a really bad sunburn on my arm, and so my wife's really angry at me now. But that process of normal tissue homeostasis and physiology, it requires ongoing cell proliferation, but it's an orderly cell proliferation where the cells divide long enough to replace the damaged tissue and then stop. Now, this is a diagram, a very simplified diagram of a mammalian cell, showing you the nucleus where the DNA is kept and where the instructions for cell behavior are encoded. And then surrounding the outside of the cell, there's a cell membrane. And in this middle bit called the cytoplasm, there's a whole bunch of functions that the cell needs to survive. But generally speaking, when a cell is required to enter into the process of cell division, it usually receives an instruction from the outside. So for example, let me just draw your attention here. You can maybe see this word growth factors hitting this thing called RTK. RTK stands for receptor, and it happens to have an activity, tyrosine kinase, RTK. When that growth factor binds to that receptor, it sets in train this process that we refer to as signal transduction. And that simply means that a signal on the outside of the cell is transmitted to information on the inside of the cell that ultimately makes its way to the nucleus and tells the cell, you need to do something. And that something you need to do is usually enter into the cell division cycle. And it could be in response to a cut, it could be in response to just normal organ turnover, it could be in response to my sunburn. But those are the signals that will rein the cell's behavior. And then once the cell has divided, maybe 1, 2, 3, however many times is required, the cell will then stop growing. However, this process goes wrong in the cancer cell. And the way we think it happens is by the subversion of the nodes in that signaling diagram that I showed you here. And I'll come back and show you specific examples of this in a second. Okay? Now, almost all cancers in human beings arise from a single cell, much the same way that we all arise from a single cell. Cancers all arise from a single cell. And so you can imagine here you have a normal cell that is undergoing a normal lifespan. And in the course of its lifespan, it might replicate and divide and make a number of daughter cells. But in the context of one of those divisions or somewhere along the line, something bad happens to that cell and it gives rise to a daughter cell that is genetically altered in some fashion. It could be in response to or in the face of, say, tobacco smoke or a dietary carcinogen or ultraviolet light exposure in the sun. But something goes wrong in that cell so that now its normal mechanisms of regulation are no longer fully intact. Now, this is not yet a full cancer cell. It's what we call an initiated cell. Something has gone wrong that makes it different from its mother. But nonetheless, this cell, having undergone that alteration, now has the capacity to expand beyond its normal limits. What we then think happens is that a second mutation builds on the first mutation, so that now the cell has additional characteristics that make it grow faster, maybe move and migrate faster, or become resistant to cell death mechanisms. A third mutation occurs, and from that group of cells, now a new population of cells that have all three alterations have now occurred. And now these cells are starting to show the properties of cancer cells. And it's the cumulative damage that builds upon itself that leads a normal cell to becoming a cancer cell. And I'll give you some specific examples of that in just a second. Now, here we're back at our cell diagram again. In the context of normal cell proliferation, sometimes you have to turn cell proliferation on and sometimes you turn cell proliferation off. And as you would expect, to turn cell proliferation on, you need things that can push the accelerator on the cell. And we know a bunch of these genes. They're all highlighted in red here. So there's a growth factor outside the cell that can tell the cell to proliferate. That's its receptor that can transmit that signal, and then components of the intracellular signal transduction pathway. In addition, there are brakes that are designed to stop the cell from proliferating. And again, in blue, here are some of the brakes. And what we've come to understand through 30 or more years of genetic analysis is that the genes that get damaged in cancer are precisely the same sets of genes that normally control the processes of normal orderly cell division. So for example, this molecule here, RAS is essential for normal cell proliferation, but if one sustains a mutation in the RAS gene, it now confers upon the cell an enhanced capacity to proliferate. In addition, a molecule, for example, like RB, is a brake on the cell. And if you combine RAS, an accelerator, with RB, which is a brake, in such a fashion that you lose the brake, you now enhance the capacity of the cell to undergo cell proliferation. And I would say that the vast majority of tumors conform to this idea of cumulative genetic damage being subject to a Darwinian-style natural selection in the human body for its capacity for fitness, is how cancer emerges, at least the primary cancers emerge. And so we classify these genes as oncogenes and tumor suppressor genes. Oncogenes are the genes that act like the accelerator on the car. They push the cell into cell proliferation, and their unrestrained activity forces the cell to continuously proliferate. However, if you keep the brakes down hard enough, even in the face of jamming your foot on the accelerator, you can still keep the car in place. But it's the loss of these tumor suppressor genes in combination with the gain of these oncogenes that now allows the proliferation of the cell to run away in an unrestrained fashion. And as I mentioned earlier on, here is an example of how this might work. If this is the sequence of DNA, which I'm sure many of you will be familiar with, it's made up of 4 key building blocks, A, C, G, and T. This instruction in the DNA is decoded through a molecule called messenger RNA, and that messenger RNA is the template upon which proteins are synthesized, and it's the proteins that do the work within the cell. So this is the information carrier, and these are the workhorses that actually do the things within the cell. And so this sequence of DNA is transcribed into a sequence of RNA, and in turn translated into a sequence of protein. However, if in this particular case, here is a sequence of the normal RAS gene. I recognize it. Here is an example of a mutation found in cancer where a single building block of the DNA has been switched. And now, instead of making the normal, nicely regulated form of RAS, a different amino acid is inserted into the protein. And now the protein is locked in the on configuration in a manner that cannot be turned off. So a single change of a single base in 3 billion human bases of DNA is sufficient to give rise to a protein that has unrestrained activity. Now, there are other ways in which one can cause mutation. In fact, many human cancers show a property that we refer to as genomic instability. So if you take a normal human cell and you do this rather lovely thing, it's called spectral karyotyping, where you can basically color the pairs of human chromosomes with different colors. So this is a normal human karyotype from a lady, 2 X chromosomes. What you can see here is that this orderly arrangement of the DNA in these pairs is often fundamentally altered in the context of a human cancer cell. So if you were to compare the same person, for example, this could be the normal tissue from a woman, and this could be the breast cancer from the same woman. You can see here that, for example, there are alterations in the structure of chromosome 5. There are 3 chromosome 7s. There are bits of another chromosome now stuck to chromosome 8, and so on and so forth. And so this process of chromosomal damage and chromosomal instability also affects genes, because the genes are on the chromosomes, and so if you lose a chromosome or gain a chromosome, now you have extra copies of chromosomes that can be making new proteins that can be behaving and contributing to the aberrant properties of the cancer cell. So there are a wide variety of different molecular mechanisms of damage to DNA, but it's the damage to the DNA that really seems to be driving the process. So, as I said, an oncogene is a gene that we believe is involved in promoting normal processes of cell replication that, when genetically altered, participates in the conversion of a normal cell into a cancer cell. And they, for a variety of historical reasons, they have a bunch of rather weird and wonderful names: RAS, BRAF, BCR-ABL, MYC, CDK4. By the time this lecture's done, you'll feel totally comfortable with all of those names. And there'll be a quiz. Tumor suppressor genes are genes that are normally involved in the suppression of cell proliferation, and that when they are silenced either by genetic alterations, genetic ablation, they can also contribute to the aberrant proliferation of the cancer cell. And so some of these molecules, like P53, you might have heard of, it's one of the most famous tumor suppressor genes, RB, ARF, and so on. And so these are the classes of genes that we think are important. So again, when you look at these in the context of the cancer cell, you see alterations in oncogenes and tumor suppressor genes that allow us then to think about ways in which we can classify individual cancers and classify subsets of cancers within the context of a large diagnosis like breast cancer or colon cancer or lung cancer. Because even though oncogenes and tumor suppressor genes are the drivers in cancer, they're different in different cancer types, and even within individual subsets of breast cancer, it's not always going to be the same oncogene or the same tumor suppressor gene that's involved in all types of breast cancer or colon cancer or lung cancer. So here's just an example of what we've come to understand about the mechanisms through which colon cancer can evolve. So, and I should say this is just one mechanism of how colon cancer can evolve. The initial mutation can cause a subtle, so this is meant to exemplify the large intestine of a human being, and then this is one zoomed in region of that section. So the single and initiating mutation can simply cause a subtle change in the cells that line the colon so that they are more able to survive under circumstances when they might otherwise die. And what forms in those circumstances is what's called a colonic polyp. And so those of you who have had a colonoscopy know that that's generally speaking what the doctors are looking for, because colonic polyps are not cancer, but they could be what we call a pre-malignant condition that if left to their own devices, they could emerge as cancer. And we know genes that are involved in this process. There's a particular gene called the APC gene, which is a tumor suppressor gene. The loss of that seems to be sufficient pretty much on its own to give rise to a benign colonic polyp. And even if you have one of these colonic polyps, it is by no means a guarantee that it will develop into colon cancer. However, if on top of that initial mutation, one layers a mutation in the gene that I mentioned, the RAS gene, now that colonic polyp changes its characteristics. And so now, instead of being just a benign polyp, becomes an aberrant crypt focus, or again, not cancer, but still not normal either. And then over the course, again, with time and with gradually accumulating genetic damage in genes like INK4, DPC4 and other molecules that I've mentioned, these lesions can gradually progress to the point where they now are carcinoma in situ in the colon. And if left long enough, they will eventually or often acquire the characteristics of being able to move from the colon to the liver. It's the major site of metastasis of colon cancer. And so the reason that we know so much about colon cancer has partly been due to the advent of colonoscopy. Because now, if a colonoscopy is conducted and a tissue specimen is removed, one can actually conduct genetic analysis on it. In addition, there are human beings that are born with a syndrome called Lynch syndrome, where it's known that there's a particular susceptibility to colon cancer that runs in the family. And those individuals are usually monitored very closely for the development of colonic polyps, and they're often removed routinely. And those discarded tissue specimens turn out to be a remarkable repository, literally a treasure trove of genetic information about the way that colon cancer works in that group of individuals, but also how colon cancer can develop in people that don't have a hereditary predisposition to the disease. Now, what is it that these oncogenes and tumor suppressors do to a normal cell that turns it into a cancer cell? So first off, we think of cancer as really having 6 cardinal properties, although this is the sort of thing that scientists love to argue that there's 9 or there's 10, but the 6 ones I think we can all agree on is that cancer cells become immortal. Those of you who read a fascinating book about a woman by the name of Henrietta Lacks, who was the person whose tumor gave rise to a cell called HeLa cells. HeLa cells have been growing in culture, established at Johns Hopkins University in 1953, I believe, and they're still maintained in culture. And cancer cells are immortal. They will grow forever as long as you supply them with a source of nutrients. They undergo unscheduled cell division, i.e. they proliferate more rapidly than they should do. They divide and give rise to many daughter cells. They are resistant to cell death, and we'll talk about this is actually an initial property of the cancer cell before you ever even see therapy. But then when you give patients cancer therapy, you can select for cancer cells that now are even more resistant to therapy because they've now been selected by the drug that's been applied to the patient. They are very resistant to anti-growth signals. They construct their own blood supply. And then most notably here is that they can invade and metastasize to distant organs. They can pick up from the colon and go to the liver. They can pick up from the skin and go to the lungs. They can pick up from the breast and they can go to the lungs or the bone marrow. And I say that's the property that is particularly lethal of the cancer cell. And as I said, just hammering this idea of the multi-stage nature of cancer, is that the accumulation of genetic damage in these oncogenes and tumor suppressor genes is the mechanism by which the cancer cells acquire these different properties, the 6 cardinal properties of the cancer cell. And they don't have to do it in a stereotypical order. It can happen in a variety of different ways, and this slide just simply illustrates the fact that the accumulation of genetic damage is very much like evolution, that the cells are constantly being selected for in whatever environment they find themselves in. They acquire a new or an altered gene function or a new mutation, and that confers upon them a selective advantage to live in that environment and outcompete their neighbors. And that's how you get selection for cells with increasing capacity for cell proliferation and to colonize the environment. Now, I'd like to point out that UCSF had a major role in understanding how this process worked, in part because of the work of these two gentlemen, Mike Bishop and Harold Varmus. I count myself fortunate as having been a postdoctoral fellow with Mike Bishop in the mid-1980s. And as you'll know, Mike was the Chancellor of UCSF for 12 years. Harold Varmus moved on, became the director of the NIH, then the head of Memorial Sloan Kettering, and is now back in government service as the director of the National Cancer Institute. And their contribution to our understanding of cancer was really quite remarkable in the sense that they were studying a very rare virus of chickens that causes cancer in chickens. And it was a matter of some conjecture as to whether cancer in chickens was really relevant to cancer in human beings or not. And I think there were a number of people who would rather have thought that it was not relevant. But nonetheless, what they discovered was that the genes that had been altered to cause cancer in chickens found counterparts in the human body, and that really opened the door to understanding and starting to catalogue the molecules that I referred to specifically as oncogenes. And this was resulted in Mike and Harold being awarded the 1989 Nobel Prize in Physiology or Medicine for the discovery of oncogenes. So what I'm going to do now is shift gears and start telling you about some specific cancers, the specific genetic abnormalities that underlie those cancers, and how our knowledge of that specific genetic damage has informed the development of drugs that make an impact in the treatment of those diseases. So first off, here's the cancer challenge. How can we exploit our knowledge of the cancer cell to devise better strategies to diagnose and treat patients with cancer? Well, so the old school way of doing this was based on just killing cells in a petri dish. And there were many, many really good drugs discovered by this mechanism. But basically how this was done is that one would identify an agent that selectively killed cancer cells in a petri dish. You'd use medicinal chemistry to turn that initial hit into a drug that could be given to people. The drug would be tested in experimental model systems, including cell cultures and animal models. Preclinical evaluation of drug toxicity to make sure that it wasn't so heinously toxic that it would kill the patient. And then this drug would then be filed as a new drug with the FDA, and then it would enter into this process that I described to you of phase 1, phase 2, and phase 3 clinical trials. And if the drug successfully had an impact on the outcome of patients with cancer, it would be approvable by the FDA, and then the hard work really started, which was to actually work out how best to use such drugs. And many of these drugs will be familiar with you, I'm sure. They fall into what we think of these days as being conventional cancer chemotherapy, right? And these are drugs that, for the most part, kill cells that are actively dividing. So, since cancer cells are actively dividing, these drugs are often very effective against cancer cells. But the toxicity associated with many of these drugs, cyclophosphamide, for example, etoposide, doxorubicin, and so on, is also associated with the fact that there are a number of other cell types in our bodies that are rapidly dividing. I mentioned our bone marrow that gives rise to the cells of the blood, the skin, the colon, hair follicles, and so on. And so the side effects associated with conventional cancer chemotherapy were largely due to the fact that when you kill growing and dividing cells, you kill normal growing and dividing cells as well as the cancer cells. And the efficacy of these drugs was dependent upon being able to find what we call a therapeutic index, or a window where you could give enough drug to kill the tumor cells and not have unacceptable toxicity on normal cells. And although that led to some very difficult side effects that had to be dealt with over many years of using these drugs, physicians have been able to work out ways in which to maximize the therapeutic benefit of agents like cyclophosphamide and minimizing, to the best of their ability, the downside of the deleterious effects. And although I'm not going to address much about drugs like this, I should say that these drugs are not going away anytime soon, and they will still remain, in many cases, the mainstay of cancer chemotherapy, in part because they have been so effective. And in my discussion of personalized cancer therapy, for any clinicians in the audience, I do not mean to disparage the use of these drugs in any way. So, the poster child for all this is a disease called chronic myelogenous leukemia, or CML. It comprises about 15 to 20% of all leukemias, and in cancer diagnosis terms, it's actually not a particularly prevalent disease, although 10 to 15,000 patients a year is by no means a small number. But if you compare that to lung cancer or breast cancer or colon cancer, where there's 150 to 175,000 new diagnoses a year, this is not a particularly common disease. And the average age of onset, which is true for many cancers, is patients who are people who are in the middle years of their life. There are 3 phases of the disease: a chronic phase, which lasts 5 to 6 years, an accelerated phase, which is 6 to 9 months, and a blast crisis phase, which is 3 to 6 months. And as you can probably tell from the way this is drawn, the chronic phase will not kill a patient for the most part. However, it's from the chronic phase of the disease that cells with more aggressive properties of growth emerge, and it's this blast crisis phase where the cancer cells are not just found in the blood, but they disseminate throughout the entire body that leads to the lethal form of CML. Now, back in the 1960s and early 70s, when the techniques for looking at chromosomes in normal and cancer cells were being developed, it was noted that patients with this disease, CML, had a very characteristic and unusual chromosome. And it was named the Philadelphia chromosome because, as you might imagine, it was discovered in Philadelphia. And that chromosome abnormality involved the exchange of information between chromosome 9 and chromosome 22. And the remarkable thing was that 95% of patients with chronic myelogenous leukemia had this chromosomal abnormality. Now, at the time when it was discovered, this was a fascinating observation and suggested mechanism, but the tools to understand the mechanism really didn't exist until the early 1980s, when it was discovered that that chromosome translocation takes 2 genes that are normally on different chromosomes and jams them together in the middle of this chromosome translocation. Those 2 genes, when joined together by this aberrant process, this chromosome translocation, leads to the synthesis of a molecule that is called BCR-ABL. BCR because that's one of the junction partners, and ABL because that's the other junction partner. This is a protein that does not exist in any way, shape, or form in any normal cell in a human being. It solely resides in the cells of the chronic myelogenous leukemia. Now, when this protein is made as a result of the aberrant chromosome translocation, it has an aberrant enzyme activity. It's known as a protein kinase, and protein kinases are enzymes that take the energy of ATP and they transfer one phosphate from the ATP onto the target protein. And this is a process that underlies the process of signal transduction, this transfer of phosphate from ATP as a high-energy donor to proteins, which then in turn modulate this function of this downstream protein. And through this process of phosphorylation leads to widespread alterations in the way in which the cell behaves, and ultimately tells the cell to enter into the cell division cycle and proliferate in an aberrant fashion. And so the idea here was, here we have an enzyme that has pockets in it that allow are required for its function as an enzyme. What if we could find a drug that gummed up the works and blocked the ability of the ATP from binding to the enzyme? If we did that, it would block the phosphotransfer reaction, and if we block the phosphotransfer reaction, maybe we would simply block the whole process entirely. And the result of that many years of research is a drug that many of you may be familiar with or have heard of called Gleevec. One of its original names was STI-571. And this is a molecule that selectively does exactly what I just described to you. It binds into the pocket on the enzyme into which ATP normally binds. I'll show you a picture of that right now. So this is what we call a crystal structure, and this is done by making crystals of the protein with the inhibitor bound and then firing X-rays at the crystals. The diffraction and scattering pattern of those X-rays allow you to reconstruct in 3 dimensions the structure of the enzyme, and in this particular case, with the inhibitor bound. So this enzyme is in pink, or red, and it has 2 regions, this region here and this region here. And the active region of the molecule where the phosphotransferase reaction occurs is where these 2 domains of the protein come together. In green, what you see here is one molecule of the Gleevec inhibitor bound into the middle of the active site. And the consequence of Gleevec binding, let me go back and forth so you can see the animation again. The consequence of Gleevec filling up the active site of this enzyme is it completely blocks the function of the enzyme in the CML cells. So all well and good. Will this work if we put it into people? So this is the very first patient with CML who received Gleevec for the disease. In the period prior to having received Gleevec, this patient received what was the conventional chemotherapy for chronic myelogenous leukemia, which is a combination of a drug called interferon and another drug called cytosine arabinoside. And what happens in this patient over the period of time is that as his white blood counts go up, indicating the presence of the leukemia, when they reach a certain level, they put him on a cycle of RSC and interferon, and they go back down again. And it cycles up and down, and you can see every time his white blood cells peaked, they'd put him on a cycle of this drug. Now, obviously, it was working for him, but the side effects of interferon and cytosine arabinoside are actually quite unpleasant. And eventually, what would have happened in this individual is that eventually he would have selected for a cell that was resistant to the drug, and he would have progressed. But you can see here that at this point, he went on the phase 1 clinical trial, the very first trial, to test the dose and concentration of drug that would be safe in this individual. And you can see here that the white blood cells in this patient went back down to the range which is normal. So anything less than 10 is regarded as being normal. And as long as he stayed on this drug, his white blood cell counts were stabilized. Now, of course, you could always argue this was an atypical patient, but it turned out he was one of many patients that were in this phase 1 study. The phase 1 study was unique in the sense that, as I told you earlier on, normally phase 1 studies are all about dose and toxicity. You rarely ever see clinical benefit. But in this particular case, they designed the clinical trial and only took CML patients that had the genetic alteration in BCR-ABL. And so in the phase 1 study, data like this was not uncommon, where this is a cohort of individuals showing elevated white blood cell counts as a consequence of the leukemia that they had. You put them on Gleevec, and now within a matter of days, it's really quite remarkable, their blood cell counts are coming back down to normal. I'm going to basically compress time here and tell you that the phase 1 clinical trial started in 1998. 53 out of 54 patients in that phase 1 clinical trial showed a response within 3 weeks, and many of the responses were durable, i.e. they lasted a long time as long as they were on drugs. Phase 1, so this was all done in patients that had the relatively indolent early stage of the disease. Similar responses were seen in patients that had the aggressive blast crisis stage of the disease. And what was found was that although these patients showed an initial response to the drug, many of them, in fact most of them, developed resistance to the drug within a very short period of time. So as you'll see in a second, this is an example where the earlier you can diagnose the disease, the more likely it is that you'll have a benefit on the patients because of the phenomenon of resistance. The phase 2 clinical trials were completed in 1999 and 2000. They confirmed the phase 1 data, and this led to FDA approval of this drug in 2001. So this is actually the fastest drug approval in FDA history. Within 3 years, the drug went from being an experimental drug to being an FDA-approved drug. We've now had this drug in the clinic long enough to have extensive experience about how useful the drug is. So what you're looking at here now is the results of a 5-year clinical trial where the drug was given to patients in the chronic phase of the disease, where they followed the outcomes in those individuals. So as you can see here, the overall survival in this patient cohort was a little bit above 80%. As you can see, if you look at all deaths, versus CML-related deaths, some people died of other diseases that were not the cancer that they were diagnosed with, right? And if you look at CML deaths, about 5% of those individuals were dying as a consequence of the recurrence and drug resistance of the disease that they had, the underlying disease, okay? What you might also notice here is that as the curves come down, there's a sort of inflection point here where it flattens out. And that suggests that if you get beyond 2 years without having become resistant to the drug, chances are you're going to remain stably in remission, because either you have the resistant cells pre-existing or you don't. And if you have the pre-existing resistant cells, that becomes apparent within 2 or 3 years. And so this suggests then that there is a remarkable effect if you get beyond, or certainly if you get beyond the next 2 years, you're in durable remission. Now, in this particular study, there was not a conventional chemotherapy arm, because by this time the drug was approved and it would be unethical to give patients a control of an inferior drug regimen. So the only way we can compare this data is to go back and look at the historical data for patients with CML treated with interferon and cytosine arabinoside. And as you can see here, you can see that Gleevec is a game changer, right? Massive difference in terms of survival, because previously all patients who would have even responded quite nicely to cytosine, riboside, and interferon would have progressed to resistant disease over the course of 5 years. Now, one of the things about this drug is that, at least up to now, we have suspected that you need to keep on taking it every single day, right? And that's because although you can massively reduce the tumour burden in patients with the disease, it would appear that you cannot eradicate, at least not yet, the tumour-initiating cells. And the reason we know this actually comes back to the question that you asked me, which is about toxic side effects. So in a few individuals that they actually developed reasonably severe myelosuppression, and so they took the patient off the drug, and lo and behold, leukemia came back, even though while they were on the drug, it was undetectable. And that suggests then that there is a reservoir of initiating cells that are resistant to the toxic effects of the drug. And we don't know yet how common that is in terms of all patients who've received the drug. However, there was a small study conducted in Italy where they gave patients Gleevec over a course of 2 years, and then they asked the patients if they would be willing to come off the drug and to see if they would show signs of recurrence. And what they found was that about 50% of the patients who came off the drug after having been on it for 2 years did not show recurrence. That suggested, and one always hesitates to use an emotive word that means a lot to people like cure, but that suggests that it might be possible to cure patients with this drug alone, or that there is a subset of individuals for which the drug could be curative. Now, I said 50% of the patients did not relapse within 2 years. To know how well that works, you'd like to follow that group of individuals for 10 years. Also, if 50% of the patients didn't relapse, 50% of the patients did relapse, and so clearly this drug's not a panacea for every single patient, so we don't yet know how to use the drug to cure patients. But this little diagram here was written in the context of a commentary on the use of the drug to potentially eradicate the tumour-initiating cells. And here what you're looking at here is the numbers of cells versus the time on treatment and the proposed mechanism of cure. So PCR is polymerase chain reaction. It's a very sensitive DNA test that can be used to look for the presence of that translocated chromosome. And in many patients, you can't even find it, even though they're on drug, right? The question then is, how much below the PCR detection threshold do you have to drive this down until you can get to the point where now there is no longer tumour-initiating cells that can sustain the disease if you take the patient off the drug? And we simply don't know the answer to that question yet. It's a matter of considerable interest from a basic science standpoint and can be modelled in the context of genetically engineered mice, where you can actually do experiments like this and follow it in a real timeline, and ultimately will be used in the clinic. Now, protein kinase is the class of molecules that I've just been talking about. There's about 518 of them in the human genome. And they're involved in every single process you can possibly imagine. And they all look the same. I mean, the details are subtle in the sense that they're not identical to one another, but nonetheless, they have a very similar fold. They all fold into roughly the same shape, and that's because they do roughly the same reaction. And it turns out that Gleevec, in addition to being a really good inhibitor of this BCR-ABL enzyme, also and completely fortuitously and by chance inhibited an enzyme similar called c-KIT and another enzyme called the PDGF receptor in exactly the same manner as it does for BCR-ABL. And it also turns out that c-KIT is important in a cancer called gastrointestinal stromal tumor and is also important in a rare set of melanomas. Similarly, there is a disease called hypereosinophilic syndrome, which is not a cancer or a leukemia, although it's thought to be a potential precursor to cancer or leukemia in certain circumstances. And again, this agent Gleevec is a very good inhibitor of the molecule that drives that process. And again, that subset of individuals with hypereosinophilic syndrome that have mutations in the PDGF receptor respond remarkably well to the drug. Now, I told you that Gleevec is best used when you catch the patients with the chronic phase of the disease, early in the disease course. I also said that in the patients who have accelerated or blast crisis disease, the drug often shows just as dramatic a remission as the chronic phase patients do. But the problem that they have is that the disease comes back in a form that is now completely resistant to the drug. And there are several mechanisms of drug resistance that occur in this context. And I'll tell you about some of those. So the first thing that can happen is you get a process called gene amplification, where the numbers of copies of the BCR-ABL gene goes from 1 to maybe 10 or 100. And if you have an increase in the copy number or the amount of the target, it's going to take an increase in the amount of drug to be able to inhibit all the molecules in the cell. And although Gleevec, compared to conventional cancer chemotherapy, is relatively non-toxic, if you push the dose high enough, you'll start to get unacceptable side effects. So then you start to run into the fact that you no longer have a therapeutic index where you can give a successful concentration of drug and block the growth of the tumor cells without having unacceptable side effects. However, the most common mechanism of all appears to be this process of second point mutations. And I'll demonstrate for you again in the context of this diagram of the structure of the enzyme. So again, this is the chemical structure of the inhibitor, the drug. This is the structure of it docked in to the active site of the enzyme. And this is a zoomed in version of that region showing Gleevec, labeled STI-571, in this kind of mauve color, showing a close contact with an amino acid that is part of the protein. And you can see how well this sort of fits like the enzyme is almost like a glove that covers up the inhibitor and binds it really tightly, which is one of the reasons it works so well. One of the most common mechanisms of resistance is where that amino acid residue here gets altered and becomes ever so slightly bigger in such a fashion that it projects its side chain into the docking site. So for those of you who know the chemistry here, the molecule starts off as being a threonine residue and by a point mutation becomes an isoleucine residue. And because the isoleucine is bigger, you can see here that you now get this clash between the drug and the enzyme in such a fashion that the drug no longer binds as tightly to the active site of the enzyme. And that is sufficient to make the enzyme 100 times less sensitive to the action of the drug. And this is a very common mechanism of drug resistance in patients with CML. That single change of a single amino acid is enough to make the protein resistant. And so what we've come to understand then is that, number one, catching the disease as early as possible in its course is important for the success of the drug. That even drugs that give dramatic effects in a more aggressive disease, that does not always translate into a survival benefit, because for those patients it did not give them a survival benefit, even though initially it seemed like the tumors were being eradicated. And that the problem of acquired drug resistance, the selection for cells that have resistant variants, that I should say are all pre-existing in the population, right? You can actually do this sensitive PCR test and show that cells that have that version of BCR-ABL pre-exist before you ever even give the patient the drug. Right? The selection for those variants is going to be a major limiting step for the use of agents like this successfully over the long term in the clinic. Okay. Right, so now I'm going to turn to a different disease, but a very similar paradigm, which is this molecule called the EGF receptor, which is found in lung cancer, and its treatment by a drug called Tarceva. So again, let's go back to this little node here. You remember I said growth factors on the outside of the cell can act by activating receptors on the surface of the cell. That's how the cell knows. I should say these slides, I added them today, and they're not in your little handout, so bear with me now for a few minutes. What we've come to understand is that when the EGF receptor is used by normal cells to signal for growth, it's found on the surface as a monomeric enzyme that actually sticks across the cell. This is outside of the cell, and this is inside of the cell. And the cell, the receptor has a hand waiting to be engaged by a ligand, and that ligand, as it's called the EGF receptor, you might imagine, the ligand is called EGF, epidermal growth factor. When the ligand comes along, what it does is it forces or promotes the receptor to form a dimer, and the dimerization of the receptor activates a whole battery of intracellular signal transduction pathways that tell the normal cells to enter into cell proliferation. When that process is done and the ligand goes away, the enzyme goes back into the ground state where it's no longer active. So that's the process of ligand-mediated receptor activation, stimulating cell proliferation, but that is then self-limiting, and the cells go back to a quiescent, non-dividing state. About 10 years ago or so, a group at Dana-Farber and a number of other institutions in Boston identified that there is a subset of patients with lung cancer with very fascinating patient demographics that remain very poorly understood. They tend to be Asian females who've never smoked. And they have a particular set of genetic alterations, damage to this gene that encodes the EGF receptor. And the consequence of those mutations is that the receptor is now active all the time, even in the absence of the ligand on the outside of the cell. So what you can think of then is, whereas before the proliferation would normally be ligand dependent to send the signal for proliferation, now the EGF receptor is on all the time and is constantly telling the cancer cells to proliferate. Genentech, in collaboration with a small company on the East Coast called Oncogene Sciences, designed, like Gleevec, a molecule which is called Tarceva, which does very much the same thing as Gleevec does, only it binds with specificity to this molecule. This molecule, as you can surmise, is a kinase. It does the same sorts of phosphotransferase reaction that BCR-ABL does. And so when you take Tarceva and you add it to patients, or cells in culture, or you give it to patients with cancer, it binds to the activated EGF receptor, and the consequence of shutting this signal off is that it promotes cancer cell death. So here's a really good example then, where knowing the genetics of the cancer and the patient demographics can predict for successful therapeutic outcomes. So nowadays in the clinic, if a young Asian female non-smoker walks in and is diagnosed with lung cancer, they're going to check this every time. In fact, overall, this comprises maybe 10% of all lung cancers. So it's not an insignificant number by any means. But this subset of cancers responds remarkably well to this drug called Tarceva. However, there are many other types of cancer, lung cancer, that don't have mutations in the EGF receptor that really don't respond at all to this drug. And there's one particular subset of lung cancers that have a mutation in the gene that I mentioned earlier on, called RAS, where the addition of this drug appears to actually make things worse. And we don't understand exactly how that works. But there's an excellent example of how patient stratification and understanding the underlying genetics of the cancer is essential to actually prescribing a drug that actually has a significant benefit to those individuals. Okay. Now I'm going to tell you very briefly about melanoma because it's a subject very dear to my heart because my lab works on it, and the gene that's important here is a gene that we've been working on for over 20 years. So melanoma is the most common cause of skin cancer death, but it's not the most common form of skin cancer. The most common forms of skin cancer are from the epithelial compartment, whereas melanoma arises from the cells that make pigment in our skin. Okay. Melanoma, for reasons that we don't completely understand, but it's almost certainly to do with exposure to ultraviolet light, is rising at a very rapid rate, and it's especially amongst the young, and particularly amongst young women. And there's some suggestion here that the use of sunbeds may be particularly problematic because of the UV dose that young women get in exposing themselves on sunbeds. This is a disease that has a very aggressive clinical course, and it has a high propensity for metastatic disease. I'm sure many of you will be familiar with the notion that metastatic melanoma has been an incredibly difficult disease to treat over the course of many years. And the treatment options for patients with metastatic melanoma have been, until very recently, extremely limited and of very limited benefit. Now, here's a slightly different way of looking at that signal transduction diagram that I've shown you before, sort of a stripped-down version. But here, if you like, is our EGF. RTK, that could be the EGF receptor. And now we're going to venture a little deeper into the cell. Here is the process and the nodes involved in a cascade of signaling enzymes that signal within the cell. And again, what these enzymes do is very similar to things I've told you about before. For example, all of these guys are protein kinases that take phosphate from ATP and stick it on another protein and change its function. And so we have this kinase signaling cascade. So what we've come to realize is that a very large subset of patients with melanoma have an alteration in this gene that's called BRAF. And again, it's a single amino acid change that changes an orderly and well-regulated enzyme into a beast that is uncontrollable, a constitutively active kinase that is constantly telling the cell to proliferate continuously. And based on the paradigms that I've told you about for CML and Gleevec, and EGF receptor in lung cancer and the drug Tarceva, companies began making drugs to try and gum up the works of this enzyme to see if this might turn out to be an effective therapy for patients with melanoma. And this is an example. Again, this is one of these high zoomed-in versions of a drug. You can see here the little ribbons of the protein with the drug docked here in the middle of the ATP binding site. Very similar in its concept to the way in which Gleevec and Tarceva work against their relevant targets. So the idea was, if we block the signaling of BRAF, that we could arrest or kill proliferation. And again, this drug has actually now turned out to be remarkably successful. Now, I should point out that the drug is not yet FDA approved, but every year, clinician scientists go to a meeting in Chicago called the American Society for Clinical Oncology, ASCO, and this was the superstar drug at that meeting. This just shows you the phase 1 clinical trials data on a small number of individuals that had melanoma and where it was documented that they had a BRAF mutation. This red line is the line that the community of medical oncologists takes as being the cutoff for a meaningful reduction in tumour size. It has to be more than 30%. And what you can see here is a very significant number of patients showed a 30% decrease in their metastatic melanoma burden. For metastatic melanoma, that was almost an unprecedented level of response, because this is the disease that the kitchen sink has been thrown at over the past 25 years, and almost nothing's had an effect. And these were large international clinical trials done at multiple different sites around the world, and the findings were very consistent from one site to the next. This drug now has gone into a phase 3 clinical trial. At the halfway point in the phase 3 clinical trial, an independent study group unblinds it, looks at the data, and they said that the patients on the new drug had gained so much benefit that they were stopping the trial and moving all the people on the conventional chemotherapy over to the experimental drug. And although this drug is not yet approved, there is every reason to believe that probably by the end of this year, that this drug will be approved as the frontline chemotherapy for patients with mutated BRAF in melanoma. And again, it's very specific, because in other classes of melanoma that don't have a BRAF mutation, the evidence suggests that this drug will not work. And so this is highly selective for a specific subset of melanoma. As it turns out, about 65% of all melanomas have this mutation. But of course, that means 35% of them don't. And so this drug will be applicable and readily useful for that subset of individuals. But again, the issues of drug resistance have already become apparent in the use of this drug as a single agent. And the fact that those of you who read the New York Times may have seen a series of articles written by Amy Harmon specifically describing the development of this drug, the clinical trials that were used to test the drug, and the fact that patients showed these remarkable responses to disease that was, they were literally at death's door in some circumstances. Within a few months, they were going back to work, but then in a few months after that, drug-resistant disease had come back, and we don't yet understand the full measure of the mechanisms of drug resistance in melanoma. So the problems of resistance are continuing to be an issue. But nonetheless, in a few slides that were presented to me, this is an imaging modality that allows clinicians to look at the metabolic activity of the cancer cell or the tumours in patients. And so what you can see here is a number of examples of individuals where they had extensive tumour burden throughout their bodies, and within a few weeks after drug administration, what you see is that a lot of the activity is gone. This is from the MD Anderson, from Vanderbilt, and from the Peter MacCallum Institute in Australia. The responses are really quite remarkable. Identifying targets, especially enzymes that drive cell proliferation, developing inhibitors that specifically target that genetic damage, and then designing clinical trials that are designed specifically to look at the consequences of that type of alteration in that type of cancer, have led to 3 successes that I've described tonight: Gleevec in CML, Tarceva in EGF receptor mutant lung cancer, and then this PLX drug in melanoma. The big problem in all of those is the resistance phenomenon that come up after, in some cases, relatively short times of treatment. And although these are all approved, well, the PLX drug not yet approved, but likely to be approved this year, as I mentioned, this is just the beginnings of the process of trying to understand how best to use these drugs in the clinic. And if you take HIV as the analogy, where it's clear that a single anti-HIV drug will result in drug-resistant virus, and that in order to minimize drug resistance, you need to apply 2 or even 3 drugs. The question now is, can you build upon the foundation stones of the success of these single agents by now adding in additional agents that will minimize or completely diminish the mechanisms of drug resistance? And that's very much an ongoing question in the basic science and the clinical science oncology community. Right, so breast cancer is obviously a very major cause of cancer in women, second only to skin cancer, but also one of the most common forms of cancer death due to the metastasis of breast cancer to lung or bone. In 2007, responsible for 41,000 deaths in the US, and it's estimated that 200,000 women will be diagnosed with breast cancer in 2010, and it's a subject very much on everyone's mind. And the general sense of it is that breast cancer obviously arises from the epithelial cells that are found in the mammary gland, but it turns out there are multiple different types of epithelial cells, and that the different subsets of breast cancer probably reflect the cell of origin in the mammary gland where this tumor cell comes from. So, using genetic tools, a number of groups have been able to subset breast cancers into about 5 different subsets. So basically, we can read out, using genetic tools, the patterns of all 20,000 genes and their expression in any specific tissue, in this particular case, breast cancer. And we can use sophisticated statistical tests to ask how similar or different are those tumors from one another. And so what you're looking at here is a sample from a paper where high-level gene expression is in red and low-level gene expression is in green. And if you ask the computer to cluster these in a manner in relation to their relatedness to one another, what you see is how they actually break up into individual clusters, suggesting that breast cancer is actually not just one disease, but in fact composed of individual subsets of disease. And it turns out that the 4 major clusters that we've come to think about now are called luminal A, luminal B, ERB2, and basal. And again, that almost certainly reflects both the cell of origin in the mammary gland that gives rise to breast cancer, as well as the types of genetic abnormality that have occurred in those cells that give rise to the cancer phenotype. And classically, these had a number of sort of old-fashioned analyses based on pathologists looking down the microscope at tissue sections taken from patients, so biopsy specimens taken from patients. But the era of molecular medicine has now allowed us to turn this into a molecularly defined set of diseases based on specific types of properties. So, for example, many of you will be familiar with the fact that the estrogen receptor is an important molecule involved in the normal homeostasis and development of the mammary gland. It turns out it's also very important in certain types of breast cancer, and the reason that women will take tamoxifen as a therapy for breast cancer is that tamoxifen blocks the action of the estrogen receptor. And so the estrogen receptor is important in this luminal A and luminal B subclass of breast cancer. And antagonists of the estrogen receptor, like tamoxifen, can be quite effective in that context. This ERB2 group, which I'll talk about in a few minutes, has a very specific gene alteration in a molecule that's very closely related to the EGF receptor. It was actually, it's called ERB2, and there's a specific therapy that targets that specific type of disease. And then these guys have a number of markers, and the disease that's now starting to become the most intractable is this basal type of breast cancer, in part because it separates itself out from the other 3, but it hasn't really been shown to have clear-cut and defining characteristics that allow us to think about ways to target it for new types of targeted therapy. And again, this just simply reflects the fact that it's thought that the breast cancers that arise in these 4 subclasses arise from different regions within the mammary epithelial structures. I told you that Human cancers often show this phenomenon of genetic instability, where the normal pattern of chromosomes is corrupted during the process of tumorigenesis. And what we've come to understand is that the luminal A and luminal B forms of breast cancer tend to be genetically more stable. If you look at the chromosomes of those cancers, they tend to look more like normal than the guys that come, or the cancers that arise or that fall in the basal or the ERB2 classification. And generally speaking, cancers that have a more normal karyotype tend to behave in a more indolent or treatable fashion. And the higher the genetic instability, generally speaking, the more likely it is that you will get aggressive variants appearing that will be life-threatening. Many of you will be familiar also with the BRCA genes, which were identified a number of years ago as being the genetic damage that underlies hereditary forms of breast cancer that run in families. They explain maybe about 15% of that inherited risk, but nonetheless, these BRCA genes are also found to be altered in the luminal A and B and ERB2 type, as well as this tumor suppressor gene P53, which is also frequently altered in this group. And as I said, this genomic instability tends to predict for bad outcome in the unstable tumours and a better outcome in the genetically stable tumours. And again, that correlates with histology grade of high-grade and low-grade tumours. And if you look here, this is a Kaplan-Meier survival curve where one is looking at the survival of patients with the different subclasses of the disease. And I think now my scale is, so luminal A is in purple, luminal B in blue, the orange is the basal, and then the kind of purple color here is ERB2. So you can see here that within this group of breast cancers, they have an intrinsically different propensity to kill the patients, and that this ERB2 group is a particularly bad actor. The basal one's next, and the luminal one's next. So the pathology analysis then indicated that breast cancer is heterogeneous, and that just simply looking down the microscope wouldn't necessarily be enough to be able to predict the behavior of the individual cancers. But the more detailed molecular analysis of the genetic damage that occurred in these cancers revealed a level of heterogeneity that was not visible just by looking at cells on a slide down the microscope. And the genetic damage that drives the cancer can be targeted by these molecularly targeted therapeutics. And what I'll tell you about now is two ways in which this can be used. The first one is to take breast cancer patients and predict who needs an aggressive course of therapy versus who needs a less aggressive course of therapy. So what was done was breast cancer patients had a small biopsy sample taken prior to any therapy, and that biopsy sample was profiled using a variety of genetic markers and a number of other criteria. Subsequent treatment of those individuals allowed the analysis to be conducted where you can relate the genetic markers and the other markers to the ultimate therapy response and the outcome in such a fashion that you could define a low-risk group of individuals that got little benefit from aggressive chemotherapy and a high-risk group of individuals that got a large benefit from chemotherapy. These patients could then be optimized for local therapy and hormonal therapy. These patients go into a much more aggressive regimen of chemotherapy. Now, if your test is sufficiently strong, it means that when a woman is diagnosed with breast cancer and you apply the test, you can predict with some reasonable degree of certainty whether that woman is going to fall into this group or this group, which means that these individuals are spared this chemotherapy. And this has led to a number of tests. The one specifically I'm going to address, and I don't mean to imply any personal analysis or vested interest on my part. I just use this one because it's one that is used at UCSF. But this is called Oncotype DX, where the expression of a series of genes and a number of markers is used to stratify patients based on this perception of risk. It's a validated test and approved by the FDA, whereby the application of these tests gives rise to what's called a recurrence score. And the recurrence score means how likely is it that breast cancer will recur in me at some future point. Okay? So A recurrence score in excess of 30 is regarded as high risk, lower than 18 is low risk, and intermediate risk between 18 and 30. And so what happens now is that low-risk individuals can choose not to have chemotherapy, aggressive chemotherapy, whereas those patients that have the high-risk Oncotype DX test are recommended to have aggressive chemotherapy. And so what that has done is allowed stratification of patients based on a perception of risk into different therapy regimens that are tailored to their specific disease. And what it's done is it has led to women that have a low recurrence score being spared from the worst of the aggressive chemotherapy that's used in patients that have a high recurrence score. Now, of course, this is still an ongoing process, and these tests are relatively new. And so what happens then when the recurrence score is 30? What's the chance of recurrence within 10 years? And again, that process is still very much being evaluated. One can say here, based on the recurrence score, that the chance of recurrence at 10 years is maybe 20%. So again, we're dealing with statistical risk here, but at the same time, it allows women to make an informed choice about the class of chemotherapy or the strategy they want to use to treat their disease. Now, so I'm going to tell you about the application of Herceptin, which is a therapy for a subset of patients with the ERBB2 positive form of breast cancer, and how that was developed. You remember, like the EGF receptor, which is a transmembrane molecule involved in receiving signals on the outside of the cell and turning that information on the outside of the cell onto information on the inside of the cell that tells the cell to undergo cell proliferation. There's a very close cousin of the EGF receptor called ERBB2, which is amplified and overexpressed in that subset of patients with breast cancer that I referred to. One of the 4 subclasses is the ERBB2 subclass. Because this is sticking outside of the cell, one could imagine harnessing components of the immune system to recognize cells that have elevated levels of this on the surface of the cell and to target those cells for destruction. And so a number of years ago, Genentech, so the idea being then that this is a driver of cancer cell proliferation when it's constitutively expressed and is constantly driving proliferation. In collaboration with UCLA, Genentech developed an antibody which is called Herceptin that binds to this region of the protein that sticks outside of the cell. The consequence of that is that the antibody, in ways that we don't fully understand, can interrupt the signaling of this molecule within the cell, but most likely the antibody serves to recruit immune cells into the local site. And the combination between the direct cytotoxic effects of the antibody on the tumor cell and the recruitment of the immune cells leads to a very significant benefit in patients that have the ERBB2 positive type of breast cancer. So here again is another example of the genetic information driving the development of new drugs and then the application of those new drugs in the clinic to actually give significant patient benefit. Now, a couple of important points about this, really important practical points, relate to the statistics of patient selection. So the frequency of ERBB2 positive breast cancer in breast cancer as a whole is about 10 to 15%. Okay? So if you were to do a clinical trial on all comers with a 10% mutation rate, you predict that only 10% of your population are going to respond, the other 90% are not going to show any response, right? However, if you pre-select for patients that already have the mutation, right, now you've pre-selected for a patient population that you predict all should have a patient benefit. And this has enormous implications in the way and the conduct of clinical trials and the timelines involved. So, just to give you an example, assuming that the expected benefit on survival is 5 months, right, if 25% of the patients had ERBB2 and the other 75% did not, i.e. an unselected patient population, what one would actually have observed in the whole patient population at large would have been a 1.25-month increase in survival, and it would have taken 11,000 patients and 350 months to conduct that clinical trial. You can imagine the costs and the time associated with that. And of course, 1.25 months doesn't look like much of a benefit. Would that even qualify as being an approvable drug? However, when you take all the ERB2 patients and you make them the sole patient group, the actual benefit observed is 5 months. It took 1,250 patients, and it took 52 months for that clinical trial to be established. So here is a drug that might otherwise have failed in the clinic were it not for the patient stratification process. But when you do the clinical trials and you focus on the relevant disease group, now you go from a clinical trial that would have been almost impossible to conduct over an extensive timeline to something that has actually had a significant impact. And again, the nature of clinical trials is that generally speaking, they're done on the sickest individuals because at that point they have expended all the conventional chemotherapies. What Genentech subsequently went back and did was they did what's called an adjuvant study. They, instead of giving it to patients once they had actually relapsed with metastatic disease, they gave it to patients immediately after their surgery to remove their breast cancer. And they did it in combination with conventional chemotherapy. And when you give Herceptin in that context, it's a game changer with regards to the potential for future recurrence of the disease. So again, one has to always be cautious about criticisms of cancer drugs that are only shown to show minimal benefit in patients, because the minimal benefit is usually always demonstrated in the sickest group of individuals. But once the drug's approved, as I emphasized, you can go back and conduct more extensive clinical trials in earlier stage patients, and the benefit can actually be much, much more significant. And Herceptin is a very good example of that process. 2 drugs have been approved which harness the power of the immune system to treat cancer. This has been an idea that's been around in cancer therapy for easily 30 or more years, and it has been largely unsuccessful until now. And in the course of the past 18 months, a drug from a small biotech company in Seattle called Provenge, which is an active vaccination strategy for prostate cancer. And then recently, a drug from Bristol Myers Squibb called Yervoy, which also mobilizes the immune system. And I think after 25 years of frustration in the development of immune-based therapies to treat cancer, we have really we've made the first mark in terms of mobilizing the immune system for treating patients with cancer. And so I think one of the things that we're going to see going forward is new approaches to vaccinate and immobilize the immune system to tackle cancers. It's still, the science is rock solid. It's the application and translation to the clinic which has been problematic, in part because I think it just is much more complicated once you get into the human to do these types of therapies. But nonetheless, these agents give hope that regulation and control or immobilization of the immune system may actually be very powerful. And what I think is going to happen is that, for example, in a patient that has a BRAF mutated melanoma, you give them a drug that targets BRAF, but we know that clinical resistance to the drug will appear because there are resistant cells that may be relatively rare. But if you could then back up the effects of the pharmacologic agent by somehow vaccinating the patient against their melanoma when they've got minimal residual disease, that you might then be able to control the prevalence of resistant cells that emerge or that would otherwise emerge in that patient. That's going to require interactions between private sector organizations that might not normally be predisposed to collaborate with one another, and it will also involve larger, important research that will go on in the public universities to understand and make suggestions about the relevant approaches that could be tested in the clinical arena. But nonetheless, I'm actually very excited about these as mechanisms or new ways going forward. I hope today that I've been able to convince you, or at least give you a sense of the excitement that lives within the cancer research community and in the clinical oncology community about the ways ahead for treating cancer. It is by no means a guaranteed success that this is going to work for all diseases, and it certainly needs the continued investment in the National Cancer Institute and the funding agencies that make the research go forward, that push the boundaries of how we understand the process and the disease and actually come up with new ways to treat cancer. And so with that, I'll close almost on time, and if there's any final questions, I'll be happy to answer them.